Hi students, I am Dr. Sagar Kumar, MSc PhD. I am having 32 years experience in the botany teaching. Today we are going to see in this video glycolysis. Glycolysis is the way where the means the glucose will be splitted. That meaning is also given. That word came from the Greek. That is the glycose means sugar, lysis means split. So that sugar will be splitted in the particular way and releases some energy that will be discussed in this one. This scheme is given by the three scientists. How it will release in the which in the particular pathway. They are the Emden, Meyerhoff, and Parnass. All these three people, they given the pathway how they will go on. This pathway will go on, and glucose will be partially oxidized and releases the energy. So on their name only, it has given the EMP pathway also. Now this is whenever we are seeing the glycolysis, it is glucose will be splitted into three carbon compounds in this way by partial oxidation. Partial oxidation means not completely oxidized, and energy releasing also partially means little bit energy only will be released. That too you have to remember one thing: without oxygen. So oxygen, if it is really doing, that is aerobic respiration. Without oxygen, anaerobic respiration. So this is will be done either aerobic or anaerobic respirative or all organism, means all living organisms will follow this glycolysis, first stage of the respiration. In this one, whenever we are seeing this glycolysis is common for all living organisms, and whenever we are seeing here the anaerobic respirative organisms are having only one this stage only will provide the energy. It won't go because the anaerobic means oxygen is required. Oxygen is not present, so it is not anaerobic respiration will do the glycolysis. That only we told all the living organisms will follow. Aerobic will go to the complete oxidation. That we'll see later on. Now in this one, whenever we are seeing in the plants, this topic name is this chapter name is also respiration of the plants. In the plants, how the glucose will get? Now glucose is going to split. How it will get? It is derived from the sucrose. Why? Because the plant used to supply to all cells its food material in the form of sucrose, as it is non-reducing sugar. Non-reducing means it won't react with any other compounds which are present in the phloem. For that only sucrose it is sending. And here they are given one statement: the sucrose is the final end product of the photosynthesis, but it is wrong. End product of the photosynthesis is the glucose, but the glucose will be immediately converted into the sucrose as it has to supply to other cells. That is the thing. So the sucrose will come directly from the photosynthesis prepared or from the storage organs also. Means whenever the more food is there, it will be stored in somewhere, and the carbohydrates also will be converted to the sucrose and it will be supplied. That all she has given in the textbook. Now sucrose, it is derived by the plant and to the cell as a food material, respiratory substrate, and after reaching into the cell, it will, this is a sucrose is a disaccharide. It will be splitted into the two monosaccharides, which are the glucose and fructose. Monosaccharides, what are? Glucose and fructose are coming from the sucrose. In the enzyme name is the invertase is the enzyme. In the presence of the invertase enzyme, the sucrose will be splitted into two monosaccharides, that is the glucose and fructose. Now, here you have to observe one thing. That is in the NCERT book, he has given the glucose only. But from where it is derived and which are coming. This also must be shown. That the importance you will come to know after seeing this video. Now you see glucose, it is stable molecule, sugar molecule, monosaccharide, and now it is going to split. So whenever it want to split, a stable molecule we want to split means we have to energize it. Energy must be given, then only it will be over energized, it will become unstable and it will be split. That is the purpose of the glucose, is taking the ATP in the first step, energizing the glucose. And this is how it will be done in the hexokinase enzyme. Will do the glucose into converted into glucose 6-phosphate, and here one ATP will be taken, and next the glucose 6-phosphate will be converted into the fructose 6-phosphate in the presence of a phosphohexoisomerase. Means it is isomerase. Why it is calling? Glucose and fructose both are the same formula, but structure is different. So it is to isomerize. Why? Because it is going to break the molecule. So structurally it must be facilitated to break. For that only it is converting to the fructose 6-phosphate. Now you have to observe one thing. He has given in the textbook also. So glucose, by taking ATP, it is converting to the glucose 6-phosphate and it is isomerized by the phosphohexoisomerase into the form the fructose 6-phosphate. And when our glucose is entering like this, fructose also will enter into the glycolysis. That he has given clearly. Directly fructose will take one ATP and forms fructose 6-phosphate in the presence of the hexokinase. That there also enzyme is the hexokinase. But here, this whatever the pathway is running, this is entire pathway is 10 reactions, chain of 10 reactions. Now here one neat bit is there, for that I am telling. If it is started from the glucose to pyruvic acid, all the numbers I given, 10 steps came. But if it is fructose, it is coming, it is 9 steps only. It will have the 9 steps containing chain reaction. This is the neat bit. In this way only he will touch. So how it will enter? When the glucose is running like this and going, it is going like this, fructose also will come and add. Means radially he is telling. Means immediately when it is going on, this also will come. Then one more sucrose molecule will be divided, glucose and fructose. So one is running like this, second one also will come and follow this. This also you should remember. So fructose will take the ATP in the presence of the hexokinase, directly it will form the fructose 6-phosphate. So fructose 6-phosphate, again it will take one more ATP. Again it will be energized for the to bring the unstability of the molecule. That is the phosphofructokinase enzyme. What it will do? It will take one more ATP and it will be converted to fructose 1,6-bisphosphate. It will be converted fructose 1,6-bisphosphate. Here whatever the 1,6 or the 6 means, in that particular carbon phosphate is attached. All these are the 6 carbon compounds up to now. Glucose 6-phosphate, fructose 6-phosphate, and fructose 1,6-bisphosphate. All are the 6 carbon compounds only. Now it is up to here, all these 6 carbon compounds are formed by taking of the 2 ATPs in different positions. This he asked once. That is in the 2023 has the ATP utilized in 2 steps assertion. Reason is the places he has given. Means where it is. It is taken when the glucose is converting into the glucose 6-phosphate, and fructose 6-phosphate when it is converting into the fructose 1,6-bisphosphate. That only. Assertion and reason both are correct. And next he has given 2019, one more question. Glucose will be converted into the glucose 6-phosphate. What is the enzyme name which is to in the reaction? That is the hexokinase. See how the bits are coming. And now it may come. That is the fructose is entering in this one. Glycolysis contains of the 10 reactions, chain reactions. How many reactions it will follow? 9 only, not 10. You can give that the statement also. So questions may arise from anywhere, any angle. So you listen this lesson properly and see the video. So fructose 1,6-biphosphate is formed by the taking of the 2 ATPs in this. So it is what it is doing, it is unstabling the glucose molecule by different modifications, and now it is energized. It will be splitted into 3 carbon compounds, 6 carbon compound C6H12O6 is the glucose. It will be converted into the 3 carbon compounds. One is the triose phosphate, 2 triose phosphates. One is glyceraldehyde 3-phosphate, G3P, glyceraldehyde 3-phosphate. And another molecule name is dihydroxyestone phosphate, DHAP. Both are having the same formula, but the structural difference is there. Remember it. And these two are formed from fructose 1,6-bisphosphate by the enzyme through the enzyme aldolase enzyme. Here remember one thing. Here I given 2 colors. Here the 2 enzyme names I given black color. But all the enzyme names I given green color. These whatever the green color enzyme names I given, it is not necessary to remember because it is not in the textbook. But it may be useful for you for the better understanding. That's all. If you are not reading those enzymes also, no problem. Because NCERT book only, whatever he has given, he used to ask bits in the Neet from that only. For example, I told already it given in the 2019. This time he may ask this one, but he won't ask all these enzymes. Now aldolase enzyme, what you do, fructose 1,6-bisphosphate will be splitted into 3 carbon compounds, triose phosphates. Those are the glyceraldehyde 3-phosphate and dihydroxyestone phosphate, DHAP. Both are formed from the fructose 1,6-phosphate by bisphosphate. They are reversible reaction. Means it can be converted this one, that may be converted this one very easily, because both are having the same formula, but structural difference. They will adjust the structure and they will become one to another. Now this glyceraldehyde 3-phosphate also called PGAL. This also you should remember, because he has given PGAL name in the text, and in the chart he has given the G3P. Such type of things he will catch in the Neet exam. So glyceraldehyde 3-phosphate or PGAL, both are the same, remember it. So glyceraldehyde 3-phosphate, whatever is there, which is the 3 carbon compound, now it will be oxidized. Only one oxidation is present in the entire glycolysis, that is glyceraldehyde 3-phosphate will be oxidized. This is the most important for the Neet. How many oxidations are there? Only one. So in the textbook he has given, that is glyceraldehyde 3-phosphate will be oxidized and releases the hydrogen, and it will be taken by the NAD, and NAD will be reduced to NADH plus. This process redox potential. Redox means reduction and oxidation. Which is oxidized? Glyceraldehyde 3-phosphate is oxidized, and which is reduced? NAD is reduced. Remember it, it is most important point. And at the same time, here it has been phosphorylized by taking the one inorganic phosphate. The inorganic phosphate will be taken and giving out the 2 hydrogens. Redox potential in the form of the 2 hydrogens also he wrote. The 2 hydrogens will come, NAD will be reduced by taking those things, NADH plus. Now, up to now, how many phosphorylation has taken place? That also need question. One, this is the When the glucose is converting into the glucose 6-phosphate, here it is phosphorylation 1. And fructose converted into the fructose 1,6-bisphosphate, here also ATP taken phosphorylation. These phosphorylations taken place by through the ATP, but here the phosphorylation taken by not through the ATP. This also it question. See how many questions you can ask, and that also you wrote in the book only. I am telling. Now this whatever after oxidizing the glyceraldehyde 3-phosphate into by oxidizing, it will give the 2 hydrogens by the redox potentials. Hydrogen will be taken by the NAD and it will be converted into NADH plus H. And for this entire thing, and it will be taken one more inorganic phosphate also. PI means inorganic phosphate. And this will be, this entire thing, reaction will be controlled by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Because hydrogens are removing from this one, glyceraldehyde 3-phosphate is dehydrogenazing. So that is the enzyme, not necessary to, I told already. The names are enzyme names, except that one, all are not necessary to you. Now, what happened? This it has already having glyceraldehyde 3-phosphate. 1 phosphate is here. 1 phosphate is added. So it will become 2-phosphate. So that is 1,3-bisphosphoglyceric acid will form. Because hydrogens are lost, phosphate is added. So new compound will be formed. That is 1,3, 1 position, 3rd position, phosphate will be there. And 1,3-bisphosphoglyceric acid is to form. That is called as a BPGA. PGA, that is 1,3-BPGA. That also given in the textbook. What you can give? The BPGA is formed means you should not confuse, because in the textbook he has given. Such type of things only you will catch, remember it. Biphosphogyceric acid will be formed from the glyceraldehyde 3-phosphate after giving the 2 hydrogens. By oxidizing, it will give 2 hydrogens. Those will be catch by reducing the, they will reduce the NAD to NADH, and it will take 1 more phosphate also. That reaction, entire reaction, will give rise 1,3-bisphosphate. That is the called as a 1,3-bisphosphoglyceric acid. Now, bisphosphoglyceric acid will lose 1 phosphate. When how it will lose? By forming the ATP. Bisphosphoglyceric acid is energetic. That energy it will lose by sending out 1 phosphate. That phosphate will convert ADP to ATP. This is the thing. So first ATP is formed. He will ask. That is the biphosphoglyceric acid is to becomes a phosphoglyceric acid, triose phosphate, that is a phosphoglyceric acid whenever it is forming at the time only. Next, whenever you are seeing, this will be done by the phosphoglycerokinase enzyme. Next, if you see here, by 1,3-bisphosphoglyceric acid become the, by losing 1 phosphate, it will become 3-phosphoglyceric acid, PGA. That is the thing. And now the 3-phosphoglyceric acid, what it will do? It will be mitase. Isomerase, mitase difference you should know. Isomerase means formulas are same, structures are different. But mitase means structure is the same, only the structure, position of the phosphate will be changed. 3 position to 2 position. Why it is doing? It is facilitating for a further reaction. So that is the thing. Now what it will do? 3-phosphoglyceric acid will become 2-phosphoglyceric acid means 3 position to 2 position it came. And all these whatever are forming, all are the 3 carbon compounds, remember it. And now 2-phosphoglyceric acid, whatever it came, it will enzyme name is the phosphoglyceromutase, because it is called as mutation. So mitase, isomerase, mutation almost same, but they are the different structural molecules. Here the same molecule. That is the difference. Now 2-phosphoglycerate, what it will do? It will be dehydrogenased. Dehydrogenase means 1 water molecule, it will send out and it will form the phosphoenol pyruvate by the enzyme name enolase. Enolase is an enzyme which is to convert 2-phosphoglyceric acid by losing one water molecule from this one, and it will become phosphoenol pyruvate. PEP. Remember, the PEP name came in the C4 plants. First carbon fixing compound is the compound that phosphoenol pyruvate, the same. Because why I am telling, you will remember here and there also. Easy to remember. Now, the phosphoenol pyruvate will lose one phosphate and become phosphoenol pyruvate. Phosphate is present. So phosphate will send out and become the pyruvic acid. And phosphoenol pyruvate, what is the pyruvate kinase enzyme? What it will do? The phosphorus it will lose by converting ADP to ATP. So phosphoglyceric acid is formed. Now all of you people are getting doubt. Why he has given 2 always? That I am going to clarify. Now this 3 phosphoglyceride 3 phosphate is running like this. Immediately it will become glyceride 3 phosphate. It also will run. Means one by one it is to go. Here also it will run. Same will come. Here also it will run in the way. This also will be converted. This one and it will come. For that only here reversible reaction. And one more. Some people are telling 7 reversible reactions are present in this one. Okay. Is it in the textbook? Not necessary. That also I will tell. First reaction irreversible, third reaction irreversible, and 10th reaction is irreversible. It is not necessary to know also, but all are reversible. You may get one more doubt. Why these many reversible reactions are there? Reversible reactions are present because the energy related reactions. If the energy is more, entire damage will be damaged. Electricity, whenever it is coming from main transform and before that one, whatever this is, like this, it is stepwise. It is to come directly. It comes. Everything will be out. For that only reversible reactions are more in the respiration, energy related reactions. Here we have to remember one more thing. That is where ATP is taken and where it is given. How many are taken, how many are given. Already I told here -1 is shown. Here in the 2 reactions, 2 ATP are consumed before splitting of glucose. Means glucose conversions. 2 ATP are taken. And how much ATP are produced? Now ATP are produced in the 1,3-bisphosphoglyceric acid, converting into the phosphoglyceric acid, 1 ATP produced, but it is going and this also coming, no? So 2. So 2 ATP are produced. And one more ATP where it produced, phosphoenol pyruvate converted into the pyruvic acid. There also one is produced, but second one also coming, so 2 pyruvic ATP are produced. So here 2 and here 2 ATP are produced, 4 are produced, 2 are consumed. If we ask the question, how many are produced? 4. How many are utilized? ATP utilized in the 2 steps, that one, and glycolysis provide 2 net. That is the ATP 4 as produced and 2 are utilized. So net how much gain in the form of the ATP in the glycolysis is 2 ATP. And not only this one, for the glycolysis, if this glycolysis end product whatever is there, that is the pyruvic acid, if it enter into the aerobic respiration, then at that time NADH plus, which is also called as electron carrier, that also he has 2018 NAD role in the glycolysis. That is the electron transport. He has given answer. Electron transport and hydrogen transport both are the same in some cases. So that is the answer. So what this NADH H plus will do, it will go to the mitochondria in the aerobic respiration and each used to produce 3 ATP in the electron transport. So glycolysis will provide 8 ATP in the aerobic respiration. If we do the anaerobic respiration, these NADH plus are not useful. They will be because electron transport system is not there, because the anaerobic it is, oxygen is not available, so it is they are adapted like that. So if it is going on that whatever the pyruvic acid goes to the aerobic respiration, at that time it will provide 8 ATP after removing this two also. If it is in this way, how many ATP, net ATP means 2. How many it can provide for the aerobic respiration? 8. And how many are produced? 4. How many are consumed? 2. All these things you remember, try to remember it or otherwise note down in the book. Now, up to this one, whatever we are seeing, that is that only I written there. 2 steps, 2 ATP, ATP produced in the 2 steps, 4. This glycolysis provides 2 net because 2 has lost. Glycolysis provides 8 ATP to aerobic organisms, 2 into 3, 6. That also I mentioned. Now, the pyruvic acid form as the end product of the glycolysis, 2 molecules, because it become 1 and this also came and become 2 molecules, it is formed. Now, one more thing I will tell. Now you see here, glucose is C6H12O6 and the formula of the pyruvic acid is C3H4O3. It is splitting into 2 3 carbon compounds. Why it becomes C3H4O3? Because it lost 2 hydrogens here. This is also a little bit, remember it. 2 hydrogens, because actually it has what should be? C3 is there, O3 is there, means half. H12 should become 6, but it is 4. Why it is 4? Because it lost 2 hydrogens when it is reaction is taking place. That is important one. One more. And next, in this way, whenever we are discussing, now pyruvic acid came out. Pyruvic acid will be undergoes. Pyruvic acid, what is the carbon? 3 carbons are means partially oxidized. How many oxidations? Only 1 oxidation. Still 3 carbons are present in the pyruvic acid. Their stored energy is present in between the carbon bonds. That energy also should come out. That is called as, if it is coming out, all the 3 carbon bonds also, then it is called as aerobic respiration. The pyruvic acid may enter into the aerobic respiration, or the pyruvic acid may be enter into the fermentation. Pyruvic acid, if the oxygen is not there, then the pyruvic acid which contains the energy will enter into the fermentation. Without giving any energy, fermentation will be taken place. That is the thing. In the fermentation, so many types are there, so many types of the acids used to form, but in the textbook has given 2. One is alcoholic fermentation, second one is the lactic acid fermentation. Here he has given little bit confusingly in that paragraph, but what he want to tell is alcoholic fermentation will be done by participation of 2 enzymes, which is pyruvic decarboxylase and alcohol dehydrogenase, if you want to see the textbook. And lactic acid, it will be only 1 enzyme will be participated, lactate dehydrogenase, directly lactic acid will be formed. Here, one more thing he has given. Lactic acid forms in the anaerobic condition. Aerobic condition also, lactic acid will be formed. How? Whenever in the eukaryote, whenever the animals are very much working, for example, also ours, when you are doing very much hard exercise, the muscular pain you will get, means it will be some uncomfortable pain will come. Why it is coming? Because this is the O2 is inadequate, because oxygen is supplied through the blood, but whatever the oxygen is supplied through the blood is not enough for the complete oxidation of the respiratory substance. That is the thing. So inadequate. So what it will do? It will undergo the fermentation, and acid will be produced. Lactic acid. Acid will be some burning sensation. For that only you will get the pain, and energy will be the only 2 ATP whatever produced here. That only energy for them. In the fermentation, whether the acid is formed or the alcohol is formed, whatever it may be, they will get the net energy. How much means whatever the energy came from this glycolysis, that only, or that only is telling in the fermentation. Here it is the lactate dehydrogenase. Here alcohol is fermentation. Pyruvic acid is converted into pyruvic decarboxylase, which is produce the one carbon dioxide. It will be sent out. And next, this product is not given in the textbook, but it is for understanding the understand the concept. It is important. Acetaldehyde will form, means one carbon dioxide as it lost pyruvic acid. The acetaldehyde again undergoes the alcohol dehydrogenase and ethanol will be formed. In the both the reactions, whenever the going on fermentation, lactic acid formation or this one, NADH, whatever is formed here, it become, because it cannot having, it won't go to this is anaerobic. Anaerobic, it is not useful, so it will be again retaken. That is reoxidized. How it is formed? By the oxidation of the glyceraldehyde 3-phosphate. Now it will be reoxidized, means it will send out hydrogens and it will become NAD will form. Here and here also the same thing he has written. So the hydrogens came and added to lactic acid. Now for that only, for showing the difference only, I shown here pyruvic acid is C3H4O3, but lactic acid is C3H6O3. Means 2 hydrogens came and again joined. Here they lost and become like this, and here again it given, it become like this. Understand, I think, clearly. Okay. Now come to the point. Alcohol fermentation, it is very important. Carbon dioxide he has given C2H5OH. Ethanol is formed, ethanol plus carbon dioxide he has given in the textbook, but 2 arrows he has given. In the first arrow only carbon dioxide will come out. But if he gives like that, go to the according textbook he give, but it is our concept I am telling you people. Now, why this he has given? Because carbon dioxide, 1 carbon and 2 oxygen has gone. So C2, it must be H6, and because this is the what is the H4, it will become H5 and H6 must become, because 2 hydrogens came, 5 hydrogens and 1 hydrogen. Why we are writing C2H5OH? That is important point for the Neet. Remember it. You know, in the chemistry it came already. If you are expert in the chemistry, you will understand. It contains of 1 ethanol group and 1 hydroxyl group. Otherwise, we can write it C2H6O, we can write. But we are not writing like that. C2H5OH. 2 carbon compound it is. So, what is the use? Because of this one, the yeast which is producing, which is fermenting the and producing the alcohol will be, that is 13% above the alcohol percentage form, it is hazardous, means harmful to the yeast itself. Whichever is produced, that alcohol, that only will die. Above 13%, it is going. If it is fermentation continuing, because it is enough metal is there, it cannot complete entire, because after reaching 13%, it will stop yeast. Then how it is hazardous, how it is harmful, poisonous for the yeast. Did you read anywhere? I will tell the cause. For that only I told this C2H5OH. C2H5OH, the ethyl group, whatever is there, neutral. It is a neutral. Hydroxyl group more negative charged, highly active. It is hydroxyl group. But though both are combined together, now what it will do? This ethyl alcohol, whatever is there, this hydroxyl group, whatever is present in that ethyl alcohol, will be highly reactive. It will be separated from the alcohol and it will react with yeast. What is the reaction? That also I will tell. When yeast 13% means it is okay. More than that, whenever it is coming, this alcohol will enter into the yeast body, because it is a neutral group is there. So neutral group easily can be diffused. But it is carrying one hydroxyl also. This hydroxyl ion will spoil all the enzyme proteins, and all the proteins are what? They are the sequence of the amino acids. And because of the bondings, it used to form primary, will become secondary, tertiary. So what are they? They are the bonds only that you studied in the biomolecules. So those bonds, hydrogen bonds, the tertiary structure is there, except covalent bond, all the bonds are present in that one. It will react with that one, it loses the structure. The enzyme will be spoiled. If the enzyme is there, not there, metabolic activity will be stopped. That is the thing. So in that way, it will kill the yeast. Now I am generating one more question to you. In the COVID, you are using the sanitizer. Sanitizer coming to the home, parents used to say, go and wash, means you will take that sanitizer and apply it for, you will keep it for 20 seconds and you will wash. What is present in that one? That is also nothing. Sanitizer contains of 60 to 95% of alcohol. That also you know, majority people knows. What they will do, either virus or any harmful germs are there, this will enter into the body of that one, that germs, and it will spoil the entire enzymes and other protein structures. It will die. That is the reason I am expecting this question, means skill, because it is related here. So for that only I am telling this one. And in the virus, if you see, virus is having the capsid, which is made up of proteins. That capsid will be spoiled if the alcohol touches there. What happens? That all protein will be spoiled by this hydroxyl ion, whatever is present in the alcohol. This is the thing. So see, you may think that glycolysis is easy, but how many NEET questions I generated, that out of that one, how many you know up to now, and how many you came to know now by seeing this video. So if you see this one question he asked in the fermentation 2022, that energy produced fermentation is, that already I told, that is the only 2 ATP molecules produced here net, that only the energy which is this, whatever this are doing, yeast and other things are doing. So that energy, how much it is? Less than 7%, textbook also given. Actually it is 5.2%. Actually it is 5.2%. So less than 7% is correct. That is the answer he has given. But I am expecting more questions because nowadays the paper last year it started becoming very hard. Means twisting it is coming. So whatever I teach you in this video, remember the point. If you read all the things in this one and remember the thing, not necessary to be afraid about the glycolysis. And the whatever the enzymes in between, they are not in the textbook, they won't be asked. But the enzyme once this asked, and I'm expecting this also next. Or it may be sanitizer will come, how it will work, how it will kill. People will say out of, but it is not out of. That base is here. And why it is a 13% above hazardous to the yeast? Because the hydroxyl group whatever is present, but that one only. And now, so maximum percentage of the fermentation alcohol, whenever it is alcohol, ethanol is producing, what is the percentage of that one? He will ask, once he asks, that is a 13% only, above 13% it only. Then how you will get the 100% or 99.99% alcohol? By distillation. How? 78 at 78 degrees centigrade, the alcohol, the ethanol will be boiled and fumes will come and it will be condensed. And alcohol, pure alcohol will come, ethanol will come. Whenever the ethanol is over in the container, in the distillation, water will start boiling. So immediately you will come to know by, as you have angular thermometer on that one, we will stop it. So whatever fumes came in the starting at 78 degrees, those are the alcohol, and they are condensed and form the liquid again. That will contain 99% above ethanol. This also can be asked. That is the thing. So with this, entire glycolysis is over. I think I covered many bits, whatever the neat bits and basic also. And if you like this video, please subscribe the channel, because next whatever are coming, I will give in the same way. And all the complete 2 years syllabus I am going to discuss in this way, which will be useful for the students. And concept orientation and analytical and basic, it is basic structure. Basically also, basic also I am giving for the 11th class student to understand. It will be useful, I think. Please subscribe the channel. If you like this video, please would give the like to this one. And next we will see the Krebs cycle and electron transport. One more thing, I used to love electron transport, how the people are explaining. I will explain it perfectly like this. I think I explained properly, but you are the you can decide it by seeing this one. So please see this one entire thing and note down the important points and read it properly, and the glycolysis you will become perfect. Questions are coming in the, here you see 22 one question came, 18 came, 19 came, 23 came, 24 and 25 didn't came. But now he is concentrating, already up to now he concentrated molecular base inherency and biotechnology, 5 questions, 7 questions like that. But I am thinking there is no criteria how many questions should be given from each chapter. So they may come on the physiology, plant physiology, then you will feel very bad. But so follow them, this thing, you will can give a answer for every question. Okay, thank you.